Hello everyone, welcome you all in the last session of Bioprocess Technology. So in our last few sessions, we are dealing with bioprocess technology, its implementation and application. In our last few sessions, we have already discussed upstream processing. During upstream processing, media formulation, strain improvement, strain development, those things are taken care. Now after that, the media is being sent for sterilization and bioreactor is supposed to sterilized. So we are almost at the middle of bioprocess technology where we are dealing with bioreactors. Now please let me allow to share my screen that you can see here. This is a diagrammatic representation. My cursor is on diagrammatic representation of bioreactor. These are the few photos of bioreactor which I have added. This last one, star tank bioreactor. Why I have given this photo so that you can understand how big it is, the bioreactor, how big they are. So, bioreactor is nothing but a huge vessel, nothing but a huge vessel which is been utilized for bioprocessing. Now, a fermenter is a huge vessel, or bioreactor and fermenter are synonymous. So in general, the size of the vessel h by d, like height by dimension, should be at around 3. The height is third times more than dimension. It is optimum size which can be utilized for fermentation protocol. When we use a bioreactor, 80% of the bioreactor must be filled with media and 20% should be remain unfilled or should be vacant. The place should keep vacant due for the foam formation. And the biomass up to 100 kg dry weight should be taken care, should be added over there. So these are the few important things while we will be doing any kind of fermentation in lab scale or large scale, we should take care. Now in the next slide, what is this depict? This depicts a total bioprocess or a fermentation process in a nutshell. If anyone understands this photograph in a clear manner, for them, bioprocess is no more a difficult part. Bioprocess is a combination of biology and processing technology, which has been depicted over here. You can see first it has been written. If you clearly see this picture, you can see first a stock culture is being utilized. Stock culture means the small microorganisms which are kept in laboratory, that from there, that little amount a shaker into a shaker flask, it is been added, and then from there a seed fermenter, they are being supplied to a seed fermenter for basic fermentation. So initially those three part comes under inoculum development. Like for 1,000-liter bioreactor, I cannot give a loop full of microorganism. At least 1% of the bioreactor should be the size of inoculum. That means for like in a 100 ml flask, at least I need to put 1 ml inoculum. So for million gallons and gallon bioreaction to take place, we need to put at least 1% of them as an inoculum. So to do inoculum development, separate process is being taken care. You can see it in this has been done over here. Now, media formulation. You can see here medium raw materials. So we are supposed to choose the raw material accordingly, and then we have to formulate the medium. Yes, we have to understand what is the type of microorganism is being used in this process, what is the time of fermentation it should be there. Based on that, we should create a medium, the cheapest carbon source, nitrogen source, minerals has to be added in the medium so that microorganism can utilize them properly. Along with that, you have to take care about foaming. Certain anti-foam should be added over there so that foaming can be get reduced. Yes, The antifoam agent should work in a manner that it should destroy the existing foam and it should hinder the foam formation. So in a both way, it should work. So then after making a proper medium for inoculum, or after making a proper medium for bioreaction, the medium has to be sterilized. Now, sterilization of the medium can take place in many manner. In our previous um, session, we have discussed that filtration, UV radiation, chemical filtration can be utilized as a source of media sterilization. And we have to sterilize the bioreactor too. So bioreactor also has to be sterilized. Then everything like developed medium and developed inoculum along with the sterilized medium are supposed to added into a sterilized bioreactor for whole bioprocessing. Now I'm putting my cursor on bioreactor. You can see this is the bioreactor. Yes, raw material has chosen and the media formulated. Now it is been sterilized. Then it is added. Inocula has been developed and now it is added over here. Now the medium has been added here in the bioreactor. Now bioprocess will take place. It can take 2 months, it can take 2 weeks. Based on the biochemical reaction, the bioprocess will be done and the media will be produced. Now, from that mixture, after certain time, my desirable product has to be taken out. That is also an important part that has to be taken out. So, from the culture fluid, the cell has to be separated. Like if I want the um, final product or end product, I have to separate the cell from the liquid. Then if my desirable product in the liquid part, then we have to go for these downstream processing to get the final product. So after bioreaction, these are downstream processing. So this part comes under upstream processing, then bioreaction, and then from cultural fluid to product packing, this comes under downstream processing. Okay. Downstream processing is been composed of not only one method, it is a series of different unit operation which has been performed serially to get the final good end product. Okay. Now, okay. This is a basic design of a bioreactor or a fermenter. This is a basic design. You can see it can be a steel vessel, it can be a glass vessel, and which have this kind of shape. There should be certain point from where aseptic inoculum can be added aseptically, like filtered or sterilized medium are supposed to add into the bioreactor. Then it's a shaft. It's a shaft which is been added with the head part, and along with that, a impeller is been added. Impeller is nothing but a rotor. Impeller will rotate, and this disc and all they will rotate and create a homogeneous mixture. As in case of our daily life, we use some spoon to stir the sugar in our tea. So similarly, this impeller work in a manner that they create a homogenized mixture inside a bioreactor. Okay, now this is the working level. As I have said that 80% of the bioreactor should be filled with this medium and 20% should be kept vacant for the foam formation, for the requirement of gas and all that should be kept vacant. Now this one is called baffle. Baffle is nothing but a horizontal bumper kind of thing. Like they will hinder the vortex formation. While everything will get rotate, it will create vortex. Now to break the vortex, certain barrier are been given. This barrier is called baffle, which reduce the vortex formation as much as uh, swirling, as much as vortex, then much amount of foam. Now to reduce the foam, these things are to be added. Now there is a sampling port. That means after certain time of fermentation, whether the fermentation is perfect or not, that has to be checked. So that should be like some sample has to be taken out from the bioreactor to get that. So this is that sampling port. Now sterile air line. For certain bioreaction, they require oxygen. They require oxygen or some other gas for their growth. So for them, sterile air or sterile, that gas has to be injected into the bioreactor. So there is a sterile gas line for that. We can say sterile air line. It's called sparger. And then here you can see a drain point, that is at the end of the bioreactor, at the end, the lowest point of the bioreactor, that is added with the drain point, so that the last drop of the mixture can be removed from this bioreactor. So this is a basic diagram. Along with that, certain ancillary part are also been added, like heating coil. Suppose the temperature is lowering continuously and I have to heat up the medium. Yes, heating coil is been added over there. Certain pH sensor and other sensor like pH, acidity, all these sensor are also added in this bioreactor. So it is a very primitive diagram. Many other ancillary parts are also there. Here I have written few parts of bioreactor, like top plate, and what is its purpose. It's covered, it's certain clamp, it's a vessel, feed pump, syringe pump. These are the some components of the ideal bioreactor I have listed here. And monitoring, how we are going, what are the like ancillary part. These are the like foam probe. Foam probe is been added to find out how much amount of foam has been gathered over there. PT100, it is a temperature sensor, platinum temperature sensor, which will provide us the exact idea that what is the temperature inside a bioreactor. Now pH electrodes, which sense the pH. Regarding this rheometer, that means what is the rotameter, sorry, what is the airflow inside the bioreactor. Viscosity, what is the viscosity of the medium is right now. Everything has to be monitored from the outside. So these are again certain ancillary part of bioreactor. So while choosing or creating a bioreactor, one should take care that why I am creating a bioreactor. What is the basic function of a bioreactor or a fermenter? It should provide a controlled environment for the optimum biomass or yield production. That means I'm providing this vessel so that it would create a controlled environment. Second, it should permit aseptic fermentation. That means there should be no hole or something like that from their fermentation can get contaminated, because contamination is not been allowed here. So it's, if there is any contamination, all the work will go in vain. So we cannot afford that. Next, it should provide adequate mixing and aeration. To give a good mix, good aeration, yes, agitator and aerator are being added into the bioreactor, which will give the best mixing and aeration of the medium. Next, the power consumption should be minimum. Okay, we are here, like in bioprocess, we are here for profit. Yes. So to get the profit, we have to do in a manner, we have to chalk out the protocol in a manner so that highest amount of profit can be obtained. So we are going to create the bioreactor in a manner that the power consumption should be very less or minimum. It should provide a easy and dependable method or mode of temperature control. That means we are supposed to have a easy temperature control system by just adding hot water in the surrounding or something like that. It should be there. Facility for sampling should be provided. Yes, one sampling port must be there, which will allow us to take certain sample from there, and which we can analyze, and we will come to a conclusion that yes, the fermentation is done perfectly. Now we can stop the fermentation. We can get the feed out of the bioreactor. Next, it should have a system for monitoring and regulating pH or other things, like those probes. Those probes should be there so that we can monitor what is going on inside. We can understand that if the microorganism is lacking any optimum condition, if the microorganism is lacking any optimum condition, we can provide that to it. Now, due to this heating, evaporation will occur, and it should like we are going to create the bioreactor in a manner, evaporation loss should be as low as possible. Otherwise, media will get evaporated, and we will face loss at the we will face problem inside the medium consistency, and ultimately microbes will not grow in a proper manner. It should require a minimum of labor in maintenance, cleaning, operating, and harvesting operations. So that means I would like to explain you that the bioreactor should be mm, designed in a manner so that the minimum labor should require for that cleaning operation, the slant, the size, the shape should be in a manner, a minimum effort should be given over there. And the final point is it should be suitable for a range of fermentation process. While I will be creating a bioreactor, we have to understand that it's a costly thing. It's not it's a very cheap product. It's a very costly thing. So if I want to change the production, then my bioreactor should have the capacity to work or to produce some other component too. It should be versatile. So while we will be choosing a bioreactor design, we'll think in a manner that we can we should create a bioreactor which is versatile in nature. Okay, so these are the few important point which should be taken care while we design a bioreactor. Few things are very important while we choose about bioreaction, that is aeration, agitation. So different type of agitator and aerators are available in the market for bioreaction. So those should be utilized for aeration and agitation purpose, because certain microorganisms require oxygen, other gas for its growth. So aeration is mandatory for them. So aeration and agitation plays a very important role in bioreaction. There are various types of bioreactors are there: stir tank bioreactor, bubble column bioreactor, a fluidized bed bioreactor. There are various, like for algae, we have that algae and cyanobacteria. There are certain special type of bioreactors are there known as photobioreactor. So there are various types of bioreactor are being present. For continuous bioreaction, some other type of bioreactor. For a stagnant or a series of like for a batch bioreactor, there are feed batch bioreactor, there are continuous bioreactor. There are various types of bioreactors are available in the market. So in our this session, I have tried my best to make you understand that yes, bioprocess technology has a wide range of application. As you can understand that bioreactors are like they can be used in many way. They can be used in many way. They are like some application includes in uh, wastewater treatment. Some application is in some other things like yes. Some applications are in wastewater treatment, some applications are there in pharmaceuticals. So, bioreaction and bioreactor all are very versatile in nature. So, if you can understand a bioprocess, if you can understand bioprocess in a very uh, just, just give me one moment. I will stop share this and I will share my screen. Mm. Okay. Okay. So in our last few sessions, we are discussing about bioprocessing, the art of turning science into success. So that means that how our everyday life is being enhanced with the products of bioprocessing. We have already have that idea. Now, it's a very easy subject to understand. There are aspects from biology background, there is aspect from technical background. Both of them are combined together for a bioprocess technology. In this session, we have learned that what is the application of bioprocessing, how upstream processing, bioreaction, and downstream processing takes care. Yes, some part of downstream processing has to be elaborated much more. If you join our class in our university, then it will be more easy for you to understand those part also. And I am also making some videos on that too. But upstream processing, I hope, is clear to you. Then there are types of bioreactors. So we are almost in the middle of bioprocessing. And then everything is being utilized for downstream processing to obtain or to fetch the perfect end product for which is desirable product by us, that has to be achieved at the end of downstream processing. In our classes, I hope I had make you understand about the implementation and applications of bioprocess technology, and that will like that knowledge will help you to explore the various aspects of bioprocess technology, like from pharmaceuticals and to food production to environmental solution. Bioprocess technology plays a very crucial as well as critical role in sustainable and efficient process. So learn bioprocess in a very depth, in a very like lucid manner, and try to apply the knowledge of bioprocessing in your own sector. In maybe it can be a research sector, maybe it's a job sector, so you can utilize your knowledge of bioprocess technology. Thank you, everyone. Thank you. The primary aim of this slide making is uh, to make you aware about this new style of technology, which is like which has an um, proper implementation, like which has a proper like blooming future. This topic has a blooming future in next few years, or already it has been started, that commercial purpose. And this primary aim is to harness the natural abilities of organisms, living organisms, to achieve commercial and practical objectives. So I hope that I had make you understand about this simple topic of bioprocess technology. Thank you, everyone. Thank you all. This is Dr. Arpita Das, Assistant Professor, Adamas University, Biotechnology Department. Thank you, everyone. Thank you.